When people hear the word chromosome, many times this image right here is what they have in mind. It is an X-looking chromosome, and that is what a lot of chromosomes look like at a particular moment in time, but they can also have different appearances to them, and we want to talk more about what it is that we're seeing here. So when we talk about chromosomes, chromosomes are going to be made up of two different types of molecules. They do have DNA, and that DNA would be in the form of a double helix, and then associated with that, we have lots of protein molecules. And the proteins are going to serve the purpose of organizing or organization of the DNA. So when we look at this chromosome, we do have this little pinched in area right here. This is what we refer to as a centromere. And then we also have the ends here, and these would be called the arms. And so we do have 4 arms on this chromosome. This would be an arm, this would be another arm over here, and then we would have 2 more on the bottom. And then if we talk about the very tips of the chromosome, these are going to be what we call telomeres. So that's the general structure of the chromosome. Now, if we dive a little bit deeper and we talk about how we get to this point, how we go from actual DNA up to this X-looking chromosome. Here we have a look at a picture of a DNA double helix. DNA itself is a very narrow molecule, but if we talk about the amount of DNA that we have inside of a single nucleus, it is extremely long. So for human cells, if we just focus on human cells, within one nucleus, you have about 2 meters of DNA. So an extremely long stretch of DNA, and it seems almost impossible, or it does seem impossible, that all of that 2 meters of DNA is going to get crammed into that very, very tiny nucleus. So the way that we do this is that that DNA is going to get organized with a lot of different proteins. So the proteins that are going to be involved in this, if we just draw one here, these are going to be called histones. Okay, so histones are organizing proteins, and they will be found right there with the DNA. And there's a lot of different histones. So they tend to be designated by letter. So we have histone A, histone B, and so on. And then we're going to have a whole bunch of these histones that are going to come together, and they will form a complex. And that complex is going to be known as a nucleosome. The nucleosome itself, that is the spool. So this will be, if we draw it down here, this will be a spool for the DNA to wrap around. Okay, so we're going to start off with a double helix of DNA. We will have a bunch of histone proteins that actually come together to form this spool, which we're going to call a nucleosome. All right, so on this picture here, what we're seeing is we are having a look at our DNA double helix. So that, the black is the DNA. And then the blue here would be our nucleosomes. Now notice that the DNA is going to get wrapped around the nucleosome. This is done in a very organized fashion. So there are a specific number of times that the DNA will wrap around each nucleosome. And then we will have a little bit of area between where we just have the DNA. This is what we a lot of times call linker DNA. That's just the little section between. And then it'll get wrapped around the next nucleosome and so on. This will be what we will refer to as the first level of DNA packaging. And just in this example, where we start to wrap the DNA around the nucleosomes, that big, long 2 meters of DNA is going to get shrunk a lot. So we will have a much shorter molecule than what we had previously. Now, this is not the end of it, because this molecule is then going to get coiled up and packaged and organized even more with additional proteins. In this picture here, we can see the full scale of DNA packaging. We start over here on the left-hand side with just the DNA double helix by itself. Then the next step is going to be what we just drew, and that's where the DNA gets wrapped around those nucleosomes that are made up of histones. Next to that would be the wrapping of those even more, so they're going to coil up, and they're going to just form this really tightly coiled, what we call solenoid structure. And then that is going to just, again, keep coiling, keep looping. And so there's all these different levels of coiling and looping. And eventually we end up over here on the right-hand side with that picture that we're familiar with of a chromosome. Now, when we look at the chromosome itself, the DNA is really kind of somewhat hidden. And what I mean by that is that the DNA is really buried under so many proteins that it's a little bit difficult to see what the DNA actually says. And the cell definitely needs to see what the DNA says because that is the instructions for making everything that they might need to make. So the instructions for making proteins, which are a lot of the enzymes and the transport molecules, hormones, and so on. So what this means is that most of the time, we really wouldn't want the DNA quite so hidden. We want to be able to use the information that's there. But at the same time, we don't want the DNA all the way over on the left-hand side here, where it's just so long that it would tend to get kind of tangled up. So we're going to be somewhere between the two extremes. So if we went and we looked inside of a typical nucleus, this will be my nucleus here, the DNA will be fairly uncoiled or unwound. And it's just, to us, visually, it kind of looks like a mess there. But it is probably more kind of in this range here. Some of it will be more compact than other areas. And the more compacted areas are the areas that get used less. So if we have certain sections that contain information to make some molecule that we only need on a very rare basis, then that would be more wrapped up DNA. If we have a section that contains information to make proteins that we're going to be making all of the time, then we're going to see it more over kind of on this end of the spectrum where it's a little bit wound up, but not completely kind of empty or what we would call naked DNA that you see over here on this end.